Good evening, everyone. Welcome to another session of MBBT. I hope you're all doing well at your side, energized, fully motivated from your whole day classes, these workshops, and another activities that you're pursuing in your life. Um, it's good that we meet in the evening during this 8 p.m. and we go through these lectures and get to know what more the molecular biology techniques, more about biochemistry techniques, and getting engaging on the regular basis. And I'm very humble actually to be in touch with all over India students here, or with the help on this online platform, and able to be connect with all of you. It's a yeah very great opportunity to with all of you, because you as a students of zoology, biotechnology, genetics, all the life sciences students here, you will be becoming future scientists, future teacher, future professors, future holding the education policies, dealing with many big steps in our life. So, so you are our future actually. Yeah, you are our future, and everything is, within your own hands. So do the right steps, make the right thing at the right time, at the right place, and gradually things will open up for all of you. Yeah. So gradually more students are joining now. I wanted it to be having at least 50 to 60 students like we were having in the initial days, but gradually I can see the students' motivation is not there anymore, and they tend to seek that they will get the certificate at the end, so they might join the class later or they even don't come. It feels sad though, but it's okay. That's the future of online education. So once we have these offline classes, we used to have a very nice attendance and everything was in order. But once this online system has started, um, so there are drawbacks. Students take some advantage of it. Anyhow, so we can start now with the leftover part from the yesterday. That is DNA microarray was done. Somebody was asking me the videos. So, bache, I will share this whole presentation with you. So don't worry about videos. So everything will be shared with you. Yeah. For meantime, I can send you some videos. It will be, but if I send you now, the streaming will be slow. So I will share it at the end of the lecture because I will share this whole presentation with you. So these presentation will have these videos. So I don't might not have to send you separately all these things. So it will work within it. So don't worry. Okay. So there are around 60 slides, and we have to start with 15th one. Cool. So investigating genes with the biotechnological approaches. So as we know, cloning is playing one of the essential role at the moment for human life, for the biotechnology at the moment. So which is helping a lot of tasks from the past easy for us. So it has made not even the agriculture successful, also the your animal husbandry, animal uh, field to be also very excited and easy for us. We can grasp many things within few time. So there are therapeutic clonings which are done in order to treat some diseases, some cancers, some mutations. So we can produce identical copies of DNA of interest that we can have a gain of one function and then analyze it. Or we can create a transgenic organism in which we have one particular gene to be either increased or decreased. So we can have large amount of proteins are possible to be produced. For example, insulin. We can produce them in bacteria or yeast from a human gene, and we can also investigate the functions of proteins in cell culture models or in animal models in order to identify the test of these effects of treatments and generating functional DNA molecules for gene therapy in gene related diseases. So genetically modified organism GMOs, they are the living organisms where gene has been deleted. That means it's a knocked out. Remember the term knocked out. That means gene has been deleted. If it is added, that means transgenic organism has been one gene has been added. That is transgenic, or it is changed like point mutation. One protein molecule, one amino acid has been removed by another one, like serine, cysteine has been placed by serine. So that will be your point mutation or mutant transgene. So in order to produce them, so we have to improve our agriculture crops with resistance against vermin or have increased content of healthy proteins. Generating transgene animals. So in basic medical research, transgene animals are extremely important for understanding of disease and possible treatments. And in farming, generation of healthy animals with improved production potential. So there was one scientist. He was in Ireland. Yeah. So he has a big farm of his sheep's. So what he used to do, he used to create transgenic sheeps actually. So he changed some, altered some genes in the sheep, and then used them for his own experiments, in order to create yeah, better yield out of it. Just a second, Papa. खड़ा ही नहीं तो जाता. So how these DNA cloning with plasmid vector works like? So let's say there's a recombinant DNA technology which enables to produce large number of identical DNA molecules. So clonings are typically generated by placing a DNA fragments of interest into a vector DNA molecule which can replicate in bacterial host cells. So when a single vector containing a single DNA fragment is introduced into host cell, large number of fragments are reproduced along with the vector. So there are two common vectors that we are using, that is E. coli plasmid vectors and bacteriophage lambda vectors. So the plasmids are actually extra chromosomal self-replicating DNA molecules. So here we can see a typical vector which is having an ORI site, origin of replication, ampicillin resistance site where we put one gene for the resistance to one antibiotics, and this is the region there we can see is known as exogenous DNA which can be inserted. So this will have polylinker, that is your MCS, multiple cloning sites. So this could have multiple restriction enzymes attachment, HIN3, SPH1, PSTL, SAL1, XBL1, BAMH1, and so on. So the general procedure for cloning with a recombinant DNA plasmid vector. So you have a plasmid vector and you want some DNA fragment to be cloned. So you enzymatically insert this DNA into this plasmid vector. So you have a recombinant plasmid afterwards. Then you add this plasmid vector with the E. coli cells in the presence of calcium chloride and grow them on the nutrient agar plates containing ampicillin. Yes, and then the bacterial chromosome will start to transform with the one which will they survive. Some cells will not take this new DNA, this plasmid, so they will not survive and they die on this ampicillin plate. Then we do the independent plasmid replication, then they start to grow and then they multiply. So we have a clone with each containing copies of the same recombinant plasmid. So we have these recombinant clones within this E. coli cells, transformed E. coli cells in huge amount. Or you can also do multiple DNA fragments to want to clone into your cells. So there's four, you take 4 plasmid vectors and you have 4 DNA fragments you want to be get cloned. So you enzymatically insert these DNA fragments into the plasmid vectors and then transform your E. coli cells and select for the ampicillin resistance colonies. So each colony here represent one clone here like this blue, pink, gray and green. So each colony will be his green, purple, reddish pink and this purple color. So they all are growing separately, the one which are identical. Beyond this, restriction enzymes play a big role in this sequencing, in this cloning. So restriction enzymes, they are very site-specific DNAs which generate double-stranded breaks. So until now, more than 600 natural occurring restriction enzymes have been identified, and they recognize 4 to 6 base pair sequencing patterns. So restriction cuts are most often asymmetric, generating single-strand overhangs, and restriction cuts are less frequently and asymmetric, that is blood end. So restriction enzymes, E. coli, so unmethylated and DNAs are there. So you cleave them. So this restriction enzyme E. coli, it tends to clean from the GATCC, and as we know they are palindromic in nature. That means if you spell from the left or spell from the right, they will always be same. Anna, auto, radar, kayak, like so. So you cleave them from here, from GA here, then you will have your sticky ends, so which could be functioned separately then. So some examples of the restriction enzymes: BAMH1, SAW3A, E. coli 1, HIN3, SM1, not L1. And how we name them actually? So what we do is we take the first name from the genus, first letter from the genus, and 2 letters from the species. So BAM. And then we take the further strain, which strain does it belong, type of strain it belong. So this is H strain and type is 1. Same like this, SAU3A. E. coli strain R and the first type, E. coli 1, HIN3, SMA1, not 1, and so on. So these are the source organisms, microorganisms for the same, and they do these recognition sites. Most of them they produced sticky ends in nature, but there's also some who are producing blunt ends, This is exceptional, which is SAM1, SMA1, CCGGGGGGCCC. So restriction fragments with complementary sticky ends are ligated easily. So there's a DNA1, there's a DNA2. So this one TTA, double TAA, it will recognize mainly AATT, the A1, not the B, not the C. So complementary and base pairs. So and there will be remain will be your B and C. So we will add ATP and also DNAs in order to join them together. So we have a recombinant DNA afterwards. So we are just talking about various techniques, how it is done, how we can do multiple cloning, how the cloning step has been done, how is your plasmid vector looks like, what are the various applications of cloning. So each type we have discussed so far. So now let's have a look a cloning vector video, small video about it. Okay, there's something weird in my computer now. The videos stopped playing now. They are not playing for strange reason. No worries, I have these videos with me now.
Plasmids can be good cloning vectors because they carry an origin of replication and are therefore able to replicate independently within a cell. Most plasmids used as vectors also encode some type of selectable marker, such as the gene for resistance to ampicillin. If the host cells are ampicillin sensitive, the only host cells that can grow on a medium containing ampicillin are those that have taken up the plasmid. Vectors must also have a small sequence of base pairs that can be recognized by a restriction enzyme. When this enzyme opens the circular plasmid, foreign DNA can be incorporated. When the plasmid vector and foreign DNA are both cut with the same restriction enzyme and mixed together, not all molecules will join to form recombinants. Some vector molecules will reanneal without incorporating foreign DNA. To identify cells that contain plasmids that have incorporated foreign DNA, a second marker gene is needed on the vector. This second marker contains the restriction enzyme site within its nucleotide sequence. If foreign DNA is inserted, the second marker is inactivated. This is referred to as insertional inactivation. A common second marker is the LAC-Z gene, which codes for the enzyme beta-galactosidase. Beta-galactosidase can cleave a colorless chemical called XGAL to form a blue compound. Therefore, colonies of cells that harbor the intact vector but no new recombinant DNA can make beta-galactosidase and form a blue color in the presence of XGAL. However, colonies that contain new recombinant DNA cannot make beta-galactosidase and are white.
Yeah. We'll share these videos later with you. So then polylinkers facilitate acceleration insertion of restriction fragments into the plasmid vector. So here we can see sequence of polylinkers. So GAATTCC, GAGCTG, and so on. So here we can see there are many restriction enzymes which could cut from different levels, right? So we have to insert. So first is this is a polylinker. Then we have to insert eco-restriction fragments. So this is the plasmid vector we are having, and this is the genomic DNA. So it has these polylinkers, many sites that it cut. And then once we insert it, so it will make this polylinker to be we will remove that part and we will add our genomic DNA into it, and we will have a recombinant plasmid. Same way, in order to clone the transcriptome, so you have a complementary cDNA libraries that are prepared from your mRNAs. So we have mRNA, tRNA, ribosomal RNA, all mixture of cytoplasmic RNA, and we wanted at the end purified mRNA out of it. So how we do it? So you put a oligo DT matrix which will have all the T um, all hybridized over it, and then we mix under the hybridized condition. So over the T, mainly your mRNA having this AAA will attach to it, and the rest will not. So we wash away the rest ribosomal RNA and tRNA, and then elute this column in the low salt buffer, so that you have a purified mRNA preparation at the end. So the preparation of bacteriophage. Further, how to prepare a bacteriophage lambda cDNA library. So just a second. So we are having these mRNAs with 3-poly A tail, which are hybridized with the oligo DT primers, and then you transcribe with the RNA into the complementary DNA. So your complementary DNA will start producing here from 5' end to 3' end. It will produce completely. Then we remove the RNA with alkali and add a poly G tail. So at the end we add this GGGG tail, and so we having now a single stranded complementary DNA out of your mRNA. And then in the next step we will hybridize with the oligo DC primer. So we will have the C trimer in the opposite of GG, and then it will synthesize complementary strand till the A. So in this way, by the fifth step, you have your complete complementary DNA with double stranded. Then we protect this cDNA by methylation. So this double stranded cDNA will be methylated. CH3 will be added on both sides, right? Then afterwards, we add these E. coli linkers, GATTC, CTAG, and then ligate into the complementary DNA to the linkers on the opposite end of it, on the left and right on both strands. And then we cleave with the E. coli enzyme. So we cleave this CTTAA part, CTTAA part from here also and from here also, so that we having these sticky ends. Then we ligate these lambda arms packaged into the E. coli. So these could be now packaged in the E. coli. So we'll have now lambda complementary DNA clones. So these techniques, they look so like complicated, in the initially you see there are around 9 steps are happening. But when you do them practically in the lab, that's the reason I want to emphasize to all students of India here, that these seems the bookish knowledge that we are gaining in the international authors or national authors, it seems quite big, it seems very complicated. Oh my god, oh there's mRNA, we have to produce complementary DNA, or we have to then protect it by methylation, or we have to ligate with complementary DNA to linkers. Oh my god, we have to add E. coli. Oh my god, then we have to ligate with lambda arms. But in principle, this experiment could be done within 10 minutes. It's just pipetting. Pipetting has to be taken care. You take your mRNA, then you transcribe to complementary DNA, and then you add your hybridized DC primers. Then you add further. It has let the reaction continue. Then you add methyl groups. Then you add ligation groups, cDNA to the linkers. Then you add E. coli. So within maybe not 10 minutes, it takes some hours, but all in all, when you do these techniques practically, they do not take much time. And same hold for any techniques that we have discussed so far. So the cloning technique, with the help of my technician in the lab, we were done within 4 days. I had within 4 to 5 days, within 1 week, quite good number of proteins in my sample. Quite good number of proteins. So this is, but what if I have started that technique to do by myself at the initially, it might take 1 month. But if you have a trained person around you who know about that technique, Ask them. There's nothing wrong in asking anyone, and there's nothing wrong in asking help from anyone. The work of your 1 or 2 months could be paced up for 1 week, so why not? Yeah, we could be help to that person also in future. So never ever hesitate or feel inferior that how the other person might think that if I can ask or not. Just do ask. Either person will say no or it will say yes. Yeah, both options are there. If they say no, it's also fine. If they say yes, that's a win-win. So go ahead, do it. Then identifying, analyzing, and sequencing cloned DNA. So the most common approach to identifying a specified clones involves the screening a library by hybridization with radioactive labeled DNA or RNA probes. So labeled nucleotide probes are also used to identify specific nucleotide sequences in complex mixture of DNA or mRNA. So direct sequence is also applied for like single nucleotide sequencing or large scale sequencing, which sequence whole libraries of your DNA or complementary DNA. So this is the most famous Sanger di-deoxy method DNA sequencing. So you have 5 prime to 3 prime end, TAGC, TGA, CTC. So you start producing complementary sequence towards it, ATC, GAC, TGA, CTC, and so on. So you add your DNA polymerase to it with addition of DATP, DGTPs, DCTPs, DTTPs, and you also add DDGTPs in low concentration, right? So what you do is you have this sequence available, and you wanted to check that um, yeah, how what is the sequence it sounds like. So what we'll do that this GGTP, it will make continue to grow your sequence until there is C is, and it will stop. Yeah, it will go until there is C. So it will grow. So we will have these sequences, and we will see them under the microscope, under the sequencer, and we can predict that this sequence is made up of this much of amino acid sequence, right? So we are adding these DDGTPs, that is your di-deoxyguanine triphosphates, right? And we are also adding these DATPs, GTPs, DCTPs, DDTPs. So these are your um, nucleosides triphosphates. So this is the three phosphates group, your one sugar and one base. So this is the blue one is the unknown sequence here, and we wanted to know about it. And we have added these GTPs, so they will get attached to it and start producing sequence corresponding to it. So in this way, you have this template you want to know about, and you have added this primer and added DNA polymerase plus DNTPs of 100 micromolar. Then this DDATPs, DDGTPs, DDTPs, DDCTPs of 1 micromolar. So they will produce along it till they could grow, till they could find it. And then we denature them and separate these daughter strands by electrophoresis. So once we're done with the electrophoresis, so here we can see various sequences. Like all the G's are here, all the C's are here, all the A's are here, and all the T's are here, right? So then after seeing them, we will start to analyze from top to bottom. So this is A, then this is T, then this is G, then this is C again, and then this is T, T, and so on. So each sequence will analyze to different corresponding amino, your nucleotides, and then we will see these graphs also, green, red, black, blue, and so on. And this will make our unknown sequence to be known then. So for the same, our Sanger, Dr. Sanger Professor Sanger got a Nobel Prize winner for this, finding the sequencing of the insulin. So let's see in practical how this Sanger deoxy method is done in the lab. Okay, so they are not producing. hope it's clear with this animation now. Now taking this sequencing to the next level, that is your next generation sequencing. So which is playing a very important role in the moment. Um, your Mahajan imaging or any imaging at the moment. So they are all using the sequencing technique in order to find out any particular genetic disease, and they are using actually artificial intelligence, machine learning, all the recent computational techniques, the bioinformatics that has been prevailing in our society, so which is playing a very important role to be. Okay, my screen is not coming nice. It's not, is it? It's somehow blurred. Just a second. Yeah, it's somehow the orientation of the screen is not up to the mark. How can I make this smaller? Okay, at least screen would be now not bad as it was before. It seems to be in center now, I guess. Yes, much better. So next generation sequencing. So it's include at the moment the recent technologies such as your Roche 454, Illumina, Applied Biosystem, Helicos, Pacific Bioscience, Life Technologies, Iron Torrent. And recently, actually, so since 2004, since these technologies have been identified, these machines, the number of base pairs to the world, they got abruptly increased. So this has been like by 2008, 100 million base pairs, a billion base pairs had been discovered. And the best part is the cost, read length, speed, accuracy, preparation time, manipulation steps. On these steps, your next generation sequencing are based on. So there's a tough competition, cost versus time basically. So which is best for the cost, which is best for the money, value for money. So there's a Roche, there's Illumina, there's a Life, Pollinator, Helicos, Pacific Biosciences, many more. There are library template preparations like FragMP, solid phase with NGS chemistry. Beyond that, there are various their own um, pros and cons with them. So best one, so this is the basic differences between your next generation sequencing. So the first one that we are seeing from the chemistry point of view. So this is a Pyrus sequencing, polymerase based and ligation based. So there's a Roche, Lumina and Solid. Three basics are the famous in the market at the moment. Right. So the chemistry of Pyrus sequencing for the Roche is Pyrus sequencing, Lumina is polymer based, Solid is ligation based. The amplification that we do is emulsion PCR, bridge amp, and emulsion PCR. And the mega base pair per run they do is 100 mega base pair, 1300 mega base pair in Lumina, and Solid in 3000 mega base pairs. So time per run is 7 hours in this, 4 days in this, and 5 days in the Solid. So read length is 250 base pairs and 32 to 40 base pairs and 35 base pairs. And the price amount you can see in the total run here is $17,447. It's 8,000 something and it's 8,400. Around this 9,000 is 8,500. And per mega base pair it's 84, $5, 5.81. So if we see from the cost per mega base pairs, then it's expensive, this Roche. These are cheap, but if you see per run in total, so this seem to be most expensive. This is the cheapest. And if you see time consumption, this is taking 5 days there, 5 days, and this is done within 7 hours. So it's up to you what you want to look for, what your demand is. You can go for the same, right? So these are the Roche workflow. We are having these DNA and they got separated, single stranded, become emulsifying PCR. Then after the PTP loading, they start to glow in this. So we will discuss about each techniques, how they work in this interested video, which will not work. Of course, you will not work. So it's a 9-minute video, so sit back and enjoy this one.
Next-generation sequencing, or NGS, is a powerful platform that has enabled the sequencing of thousands to millions of DNA molecules simultaneously. This powerful tool is revolutionizing fields such as personalized medicine, genetic diseases, and clinical diagnostics by offering a high-throughput option with the capability to sequence multiple individuals at the same time. Linear sequencing, first developed in the 1900s, is a gold standard for DNA sequencing, and it is used extensively for routine sequencing applications and to validate NGS data. It utilizes a high-fidelity DNA-dependent polymerase to generate a complementary copy to a single-stranded DNA template. In each reaction, a single primer, complementary to the template, initiates a DNA synthesis reaction from its 3' end. Deoxynucleotides, or simply nucleotides, are added one after the other in a template-dependent manner. Each reaction also contains a mixture of 4 di-deoxynucleotides, one for each DNA base. These di-deoxynucleotides resemble the DNA monomers enough to allow incorporation into the growing strand. However, they differ from natural deoxynucleotides in 2 ways. 1. They lack a 3' hydroxyl group, which is required for further DNA extension, resulting in chain termination once incorporated in the DNA molecule. And 2. Each di-deoxynucleotide has a unique fluorescent dye attached to it, allowing for automatic detection of the DNA sequence. As a result, many copies of different length DNA fragments are generated in each reaction, terminated at all of the nucleotide positions of the template molecule by one of the di-deoxynucleotides.
The reaction mixtures are loaded on the sequencing machine, either manually onto slab gels or automatically with capillaries, and are electrophoresed to separate the DNA molecules by size. The DNA sequence is read through the fluorescent emission of the di-deoxy nucleotide as it flows through the gel. Modern-day Sanger sequencing instruments use capillary-based automated electrophoresis, which typically analyzes 8 to 96 sequencing reactions simultaneously. Next-generation sequencing systems have been introduced in the past decade that allow for massively parallel sequencing reactions. These systems are capable of analyzing millions or even billions of sequencing reactions at the same time. Although different machines have been developed with various differing technical details, they all share some common features. 1. Sample preparation. All next-generation sequencing platforms require a library obtained either by amplification or ligation with custom adapter sequences. 2. Sequencing machines. Each library fragment is amplified on a solid surface with covalently attached DNA linkers that hybridize the library adapters. This amplification creates clusters of DNA, each originating from a single library fragment. Each cluster will act as an individual sequencing reaction. And 3. Data output. Each machine provides the raw data at the end of the sequencing run. This raw data is a collection of DNA sequences that were generated at each cluster. The differences between the different next-generation sequencing platforms lie mainly in the technical details of the sequencing reaction and can be categorized in 4 groups: pyrosequencing, sequencing by synthesis, sequencing by ligation, and ion semiconductor sequencing. In pyrosequencing, the sequencing reaction is monitored through the release of a pyrophosphate during each nucleotide incorporation. The released pyrophosphate is used in a series of chemical reactions resulting in the generation of light. Light emission is detected by a camera which records the appropriate sequences of the cluster. The sequencing proceeds by incubating one base at a time, measuring the light emission, if any, degrading the unincorporated bases, and then the addition of another base. This technology is capable of generating large read lengths, much comparable to the read length of Sanger sequencing. However, high reagent cost and high error rate over strings of 6 or more homopolymers have reduced. For more details on the technical aspect of this technology, please visit our knowledge base at the link provided in the description below. Sequencing by synthesis utilizes the step-by-step incorporation of reversibly fluorescent and terminated nucleotides for DNA sequencing and is used by the Illumina NGS platforms. All 4 nucleotides are added to the sequencing chip at the same time, and after nucleotide incorporation, the remaining DNA bases are washed away. The fluorescent signal is read at each cluster and recorded. Both the fluorescent molecule and the terminator group are then cleaved and washed away. This process is repeated until the sequencing reaction is complete. This system is able to overcome the disadvantages of the pyrosequencing system by only incorporating a single nucleotide at a time. However, as the sequencing reaction proceeds, the error rate of the machine also increases. This is due to incomplete removal of the fluorescent signal, which leads to higher background noise levels. Our NGS, an introduction knowledge base, provides more technical details about this technology. Sequencing by ligation is different from the other 2 methods, since it does not utilize a DNA polymerase to incorporate nucleotides. Instead, it relies on 16 octamer oligonucleotide probes, each with one of 4 fluorescent dyes attached to its 5' end, that are ligated to one another. Each octamer consists of 2 probe-specific bases, and 6 degenerate bases. The sequencing reaction commences by binding of the primer to the adapter sequence and then hybridization of the appropriate probe. This hybridization of the probe is guided by the 2 probe-specific bases and upon annealing, is ligated to the primer sequence through a DNA ligase. Unbound oligonucleotides are washed away, then the signal is detected and recorded. After that, the fluorescent signal, along with the last 3 bases of the octamer probe, are cleaved, and then the next cycle commences. After approximately 7 cycles of ligation, the DNA strand is denatured and another sequencing primer, offset by 1 base from the previous primer, is used to repeat these steps. In total, 5 sequencing primers are used. The major disadvantage of this technology is the very short sequencing reads generated. Ion semiconductor sequencing utilizes the release of hydrogen ions during the sequencing reaction to detect the sequence of a cluster. Each cluster is located directly above a semiconductor transistor, which is capable of detecting changes in the pH of the solution. During nucleotide incorporation, a single hydrogen ion is released into the solution and it is detected by the semiconductor. The sequencing reaction itself proceeds similarly to pyrosequencing, but at a fraction of the cost. Please view our knowledge base for further details on ion semiconductor sequencing and the sequencing by ligation techniques. In order to be able to showcase and compare the different technical aspects of each of the above technologies, the number of coverage that each run generates when sequencing the whole human, mouse, Arabidopsis thaliana, and E. coli genomes are calculated and presented here. The presented data is based on the most powerful machines of each technology. Further details can be found on our knowledge base. For whole genome sequencing data to be useful, a minimum of 30 times coverage is required. As it can be seen, the pyrosequencing method is only able to sequence the E. coli genome at enough coverage to result in valid data. The sequencing by synthesis method, which is the most popular method currently on the market, is able to generate hundreds of coverage per run. In fact, with this machine, it is possible to sequence 15 individuals within 3.5 days. The sequencing by ligation method also generates enough coverage for all genomes to be used. However, it isn't capable of generating nearly as much output as the Illumina HiSeq machines. The ion proton machine is used mostly in clinical settings because it is able to provide a sufficient size output within 2 hours. ABM offers a wide range of next-generation sequencing services. These include whole genome sequencing, exome sequencing, RNA sequencing, disease panels, lane rentals, and much more. To be able to access our services, please visit our website at www.abmgood.com, and from there, click on the NGS Sequencing Services link. This will load our NGS service webpage, which details all of our available services. Clicking on a service of interest will showcase the technical details, pricing, and bioinformatics solutions that are related to that particular service. Please leave your questions and comments below, and we will answer them as soon as possible. For more information, please visit our knowledge base at the link provided below. Thank you for watching.
So, so I hope this next generation sequencing concept is clear today. Yeah, it's still going on. Awesome. So this one we will start tomorrow. This is about the next topic, the method to study gene functions and how your transgenic, how yours knockout, knock in, all various types of mouses, how they are created. So we'll go in detail about them from the perspective of many genotype, phenotype, many things. So today our main concern was mainly to understand that how cloning is done and how next generation sequencing, including your Sanger sequencing, are being taken care. Yeah. So I think I should not put much hard work on a single day for you. So now I can share these videos because we might not stream anymore, because with streaming the uploading of these videos become very slow. Okay, I will forward them to Okay. So once they are being uploaded, I will forward to your June batch also. Right? So till that time, take care. Yeah. Enjoy your evening. Keep learning. Keep growing, and be the star of the future. Take care. Lots of blessing. Bye bye. Yes, we will meet us tomorrow also, and we will try to finish this part of your third day of tools of molecular biology. So we will finish them tomorrow, everything. So please meet me tomorrow at same time, same place. I will also message this in the group. Yeah. So thank you. Take care. Bye bye.